our next speaker is Dr. Tracie Matsumoto. She is a research leader at PBARC, Supervisory Research Horticulturist. She's been with us since 2002, has worked on developing tissue culture and horticultural management techniques for subtropical and tropical fruit crops with an emphasis on flowering. She's been our acting curator of our germplasm repository since 2015. And she is currently our research leader for the Tropical Plant Genetic Resource and Disease Research Unit. And she's going to talk about our efforts on the genetic side and the possibility of bringing in CLR-resistant varieties into Hawaii.
Okay, well, thank you very much for the invitation, and it's a pleasure to speak with you today. So today what we're going to talk about is coffee variety testing that we're doing at PBARC. And so what I'm going to do is just to give you a brief overview. We're going to give you, I'm going to talk about the World Coffee Research International Multi-Location Trial, which is part of the USDA SCRI grant that Suzanne had mentioned earlier. We're going to go a little bit into some coffee propagation work that we're doing, talk to you a little bit about our coffee germplasm, and look at some projects associated with the coffee germplasm. And so just to give you a little bit of background, I just wanted to, actually it just occurred to me when I was putting the slide set together last week. Prior to October 2020, Hawaii was the only major coffee producing region without coffee leaf rust. And so, but thanks to Dr. Nagai at the Hawaii Ag Research Center, she actually had starting a CLR breeding project. Oh, I'm sorry. Coffee leaf rust breeding program for resistance in Hawaii. And because she had initially started these crosses, and she had developed a network of researchers worldwide, including a collaboration with Dr. Vitor Vareza from Portugal. And so he is the world authority on coffee leaf rust in determining races. And so I'm not sure if you're familiar with how you determine a coffee leaf rust race, but basically what you have to do is you have to inoculate the coffee leaf rust spores onto different genotypes of coffee. And only from piecing together which ones are susceptible, which ones are tolerant, that's how you can discover what the leaf race is. And so we're fortunate that because of this collaboration, he was able to do our race determination and found out that we do have race 24. And so it does look like it was a single introduction into the state. And so prior to 2020, in 2017, we started working with Dr. Nagai to become a part of the International Multi-Location Trial. So what is the International Multi-Location Trial, which we call IMVT for short? Okay. And so basically what it is if you notice the countries across the map, you'll see that if they have cultivars listed below them, those are the ones that they selected as their top producers that they wanted to trial around the world. And so the rest of the countries, so 22 in all, were involved in evaluating these, all of these cultivars in their collection. And so we were fortunate to get actually some of the last material that was available, and so we brought those into the lab. If you want to know more about these varieties, the World Coffee Research actually has a good database that you can see, and you can search for the varieties. And so what you can do is you can see how they grow at different altitudes, but production, the stature of the trees, as well as their cupping quality. So these are the coffee varieties that we have in tissue culture. And so these are the ones that they deem susceptible versus tolerant. I just want to do a caveat that we don't necessarily know how they will react to race 24. But in general, these are generally considered the susceptible versus tolerant. Okay, so in addition to all of the varieties that they are producing here, we are also including Typica as our standard, just because we know that this is the standard that any coffee would have to meet to be suitable for the Hawaii specialty market. And so we have, this is actually a large trial where it's set up in a Fisher block design, so it's randomized complete blocks with your tall trees and your dwarf trees. That's the data that we're getting on horticultural, evaluation, looking at disease resistance, looking at cherry phenology, production data, bean quality, as well as cupping quality. And so we do have locations around the, to include the different microclimates and the different islands in Hawaii. And so as an added benefit, we also have Dr. Roxana Myers, who will be screening the cultivars for root rot resistance to root knot nematode. And so then we would know if these varieties would need to be grafted or they could just be grown on their own. So that one is currently underway. And what we're also looking at is so these varieties were imported, they were put into quarantine for about a year where we weren't able to propagate them. And then so now we're in the process of ramping up the numbers so that we can start the coffee leaf rust trials. So in the meantime, since coffee leaf rust did hit, what we're looking at is what varieties that might be present in Hawaii. And so I believe Lisa had mentioned some of the material from HARC. Also, you might have heard about the obatas or 2P, which is 2P or not 2P or false 2P as it's called. But so those are some of the varieties that Lisa has been very actively involved in screening, and we are looking at propagating. And so this is just an example of some of the material that we found in a grower's field. And so we were able to give these to Dr. Mike Shintaku, his lab, which is propagating these through somatic embryogenesis, which is a much faster way of doing clonal propagation. And so we are also fortunate to work with Dr. Daopeng Zhang from the from USDA ARS in Beltsville. And so he was our research geneticist. And so he was able to actually, we sent the material to him, and he was actually able to genotype these. And so those three that you saw in the previous photo, we know that they have a background from Ethiopian and maybe a Sachamour background, which is one of the Timor hybrids. And then one of them, which is this taller one that actually looks more similar to the material grown here, actually has a lot of bourbon in the background. And so we are in the process, and we're hopefully this year we'll be able to get the cupping data from these. But because the propagation does take a long time, what we wanted to do is start the propagation now, so that if we found out that these are acceptable cultivars, then the material would be ready a little bit faster, because everything, takes a while. Okay, so now I'm going to move on to the germplasm. And so we were fortunate in 2019 to actually get coffee added to our repository primary crop list. And so what, I'm not sure if everyone here is familiar with the National Plant Germplasm System, but what we are is we're a series of gene banks spread across the country that what we do is we preserve important agricultural crops. And so, for an example, Geneva hosts all the different apple cultivars. In a clonal repository in Davis, they have all the walnuts and, walnuts and pecans. And so we work together as a system, kind of as a Noah's Ark. And so if you've ever heard of Svalbard, which is the doomsday vault in Sweden, where they send seeds, so in case there's ever a catastrophic event that happens, we can restart the food system again. And so the ARS does contribute to that resource as well. Okay. And so just to go over what's in the Hilo Bank, so because we're in a subtropical tropical region, we do have a lot of subtropical tropical fruit crops, including the pineapple, breadfruit, starfruit, peach palm, pili nut, papaya and its relatives. We're very happy to be adding coffee, longan, lychee, macadamia, acerola, rambutan, pulasan, passion fruit, and guava. So, okay. So the National Germplasm System, since we're starting off fresh, what we're doing is we're fortunate to be part of this larger network. And so what we do is we have the genotyping that's available, that's been developed by Dr. Zhang in Beltsville. And so he evaluates that. We import it and put it into quarantine into our facility. We will have a field collection in Hilo, and we also have a backup collection in Mayaguez. So Hilo is one of 3 subtropical tropical fruit repositories in the United States. Because if you realize we are in Hilo, Miami, and Mayaguez. And so what do we have in common? And we're all in hurricane-prone areas. So what we do is we actually will send a core collection of what we store and put it in Puerto Rico or put it in Miami. So that way, in case something catastrophic happens, we'll have that material will be preserved. So a good example of this is that because of avocado laurel wilt, in Florida, which is a disease that affects avocados, it was getting closer and closer to the station in Miami. So what they did is we started a program to actually import their whole avocado collection into Hilo. And so to do this safely, what we did is we sent the material first to Fort Detrick. They tested the material there. They got the scion, and before sending it to us here in Hilo, where we're actually propagating it and maintaining it. Okay. In addition, we also do long-term storage or looking at how to preserve this for the future. And so a good way that they do is they actually put it in cryopreservation, where they actually take a little bit of tissue and then they freeze it, and then you can store it forever. Unfortunately, working with subtropical tropical fruits, they're not so keen on being frozen, and so, so that's why it's always important to have that backup. But there are techniques, certain techniques that they can do to accomplish those goals. Okay, so to get a little bit more into the genetic characterization of the coffee germplasm, what Dr. Zhang had developed was a SNP marker-based approach where he can use a small part of DNA. And so when you see when you see a base change in that red highlighted box, then you'll see that there's a difference in diversity. And so what he's able to do is he's able to get 132 accessions from Tracie. And so he's actually genotyping their entire collection, and from there, and as well as another gene bank in the Ivory Coast, and the material in HARC, what we're able to do is find out what the core collection to make sure that we represent enough of the genetic diversity, and then have that available to us here in Hawaii. Okay. And so this is just kind of some of the examples of what he does. So what you can do is he can group them into different groups based on their genetic similarity. And so you can see he's able to separate the Katuais, Typicas from some of the other material. So some of the other projects, so we're very fortunate to have this funding and the germplasm added to as a resource. And we're also very fortunate that we have our own research geneticist who's been newly on board, so Dr. Qingyi Yu. And so together with Dr. Mingli Wang, who is, I guess, taking over the work for Dr. Nagai, they also have another SCRI project, which is looking at breeding or crossing some of the elite, some of the resistant cultivars, such as Catamore, and crossing those with elite Hawaiian cultivars, such as Typica or your Katua'is. And so that way, the thought process is that you could still preserve the Typica type of appearance, but add in the resistance gene. And thanks to the work with Dr. Qingyi, hopefully what we'll be able to do is develop markers to speed up that breeding process. And then through Dr. Lisa Keith's work, who you just heard earlier, what she's able to do is rapidly screen the material to expedite the process and selection process. So we're also a part of the Innovia Global Arabica Breeding Network, which is also in conjunction with Rural Coffee Research, and using the same principles that we're using for the Hawaii breeding trial, but this one we're looking at it worldwide. So the material that we're going to incorporate from different regions will be, they make the crosses in the other countries, they send it to us, and what we're able to do is then develop our own cultivars for the future. And basically what we want to incorporate is genes that for material that is not present in Hawaii. So right now we do have race 24, and as Lisa had mentioned, we want to maintain that we just want to have this one race, and so we don't want to develop new races, but in case they do come in, we do want to be prepared. And so we also want to be prepared for coffee berry disease, leaf miner. And so the advantage of working with these global partners is that they'll be able to test the diseases and pests that we don't necessarily have here. Okay. And so I just wanted to thank, it's so wonderful to work with so many people who are aimed towards a goal in helping the Hawaii coffee industry. And so with our collaborators at USDA, Hawaii Agriculture Research Center, World Coffee Research, and of course Suzanne, who I'm convinced does not sleep with all her projects, and we really appreciate her support. And of course, we want to thank our funding agencies, which are the SCRI grants from 2001 and 2002. And I really have to give a shout out to our tissue culture technicians, Donna Ota and Lisa Mao, who are trying to propagate the lines as we currently speak. And so, in addition, we have just a wonderful group of people to work with, and everyone's always willing to pitch in and help out. And so, for that, I am truly appreciative. So, and with that, hopefully on time.